All right, afternoon, everybody. Couple of quick things. We're gonna be lecturing today, lecturing on Monday. Tuesday, we do have a lab class. Tuesday, there is no pre-lab quiz due. There will be a content worksheet that I am behind, but I'll get that posted this afternoon. So for Tuesday, we do have a content worksheet that's due. For Tuesday, we're also going to have a lab quiz. And that's going to be the final lab quiz for the semester. So that is going to be, so it's content worksheet for Tuesday and then a lab quiz for Tuesday. And I'll email out reminders about this. The lab quiz will cover the environmental microbiology lab. That's the one where we did the soil samples, water samples, and the oral biofilm samples. It will cover the disease pathogenesis lab. That was the one where we went around shaking hands with bacterial covered hands. Oh, we didn't talk about that. Oops. All right. I was supposed to talk about that on Tuesday and I completely forgot with all the other stuff in the exam. Okay. And so maybe we'll get a chance to go through the data and see what people found from that one on Tuesday as well. And then we'll talk, and then it'll be a little bit over the evolution and antibiotic resistance lab we started this past week, but are going to finish up then on Tuesday then as well. So those plates you guys did of either wild type E. coli or the mutant E. coli, I've taken those out of the incubator and I put them in the fridge for right now. So they're just going to stay in the fridge until next week. We'll get those out next week and we'll start doing data collection as a class on that then. Okay, so that's what we're going to be doing next week. But again, we're going to have a content worksheet due Tuesday, lab quiz, final lab quiz on Tuesday, and then we'll do data analysis on that one. The other thing is it's just a lab period to keep working on your unknowns project. So that's what's coming up there. We are going to keep lecturing to the end of the semester. We kind of left off talking about passive regulation of gene expression. And passive regulation is where the regulation is built into the genetic sequences and happens on its own without any kind of outside input. So the genetic sequences themselves regulate the expression level. And there's no real control active to the process. It's just passive and it's built in. That's kind of the default of how it's going to be expressed. And we talked about how promoter consensus sequences can result in passive gene regulation, how Shine-Dalgarno sequences, the ribosomal binding site sequences, can result in passive regulation of gene expression, how much of that gene is going to get expressed. And we talked a little bit about how also codon bias can sometimes play a role in passive regulation of gene expression. Today, we're going to talk more about some active regulation of gene expression. That is some common ways that some genes are turned, in essence, on or off based upon environmental signals. So with passive regulation, it's built in, it's kind of the default. This controls the expression level. Active regulation, bacteria often will make certain proteins that can sense their environmental conditions, but then can regulate and turn genes on and off based upon those conditions. So there's certain proteins that are made only in response to particular environmental conditions. If they're in a certain environmental condition, they're turned on. In other environmental conditions, they're turned off. But when this happens, the bacteria has to have some way of sensing those environmental conditions and then changing gene expression in response to those environmental conditions. That's what I'm kind of getting at here with then what is considered active regulation. Active regulation is there's an environmental condition, the bacteria sense that, and then they change their gene expression in response to that. And it's a more active process. It's not a default or passive process, which is what we were talking about previously. I'm going to talk about some different kinds of active regulation, but these are important. Why would the bacteria change its gene expression levels in response to different environmental conditions? Kind of depends, but Isaiah?
Like It may need certain proteins if it's in low temperature environment versus its optimal temperature.
And that might be part of it. I'm good. That's a good summary, don't you think? Yeah. Okay. So they might need certain proteins to respond to certain conditions. Why not make those proteins all the time, though? If you need certain proteins to survive in cold conditions, why not always make those proteins? And if you end up in cold conditions, the cell survives the cold conditions. Why not have those cold conditions surviving proteins on all the time?
It's not very efficient.
Yeah, it's not efficient. So really, yes, that is part of it, but it's really about why do we regulate it? It's about efficiency. Make what you need and only what you need is the idea with then regulation. Why do we do this thing? Is because say you have an enzyme that's responsible for breaking down lactose as a food source. And we're going to talk about an example of that with the lac operon. But if those enzymes, their only job is processing lactose, would it make sense to express those enzymes if no lactose is around? No, because you'd be making those proteins and it costs a lot of energy to make those enzymes, to make those proteins, and then they'd just be sitting around doing nothing if there's no lactose around. So all of these regulation things are a way of improving the efficiency of cells, of saving resources or energy for the cells then. And so that's why we're going to go through some of these. There are different active regulation strategies that I'm going to talk about. First one we're going to talk about is alternative sigma factors. And this controls transcriptional level. It turns transcription on and off for a particular gene. We're going to talk about some DNA binding proteins that control then gene expression. We're going to talk about some specific examples, lac operon, two-component systems, and quorum sensing. But again, this controls gene expression at a transcriptional level. And when I say it's controlling it at the transcriptional level, do you make messenger RNA off that gene or not? You're controlling it at the level of transcription. Does the RNA get expressed from a gene sequence or not? But we're also going to talk about some types of translational regulation. This is going to control, does a messenger RNA get translated into a protein or not? So some control mechanisms are at the transcriptional level. Some control mechanisms are at the translational level. So a gene does get expressed into messenger RNA, but maybe not into protein in some cases. When we talk about things like anti-terminators, riboswitches, and small RNAs, and their role in active regulation of genes. Right, first one we're going to go through is alternative sigma factors. What's a sigma factor? We've talked about that previously. Yeah. So promoters are DNA sequences, and promoters are where we start what process? Transcription. So promoters are DNA sequences where you start transcription. But in prokaryotes, what ends up happening is you have a promoter, a DNA sequence, sigma factors come in and bind to that promoter. Then the sigma factors load the rest of the RNA polymerase holoenzyme onto there, and then they do transcription. They make the messenger RNA. We talked about sigma factors earlier on when I went through sigma A, or what's called the housekeeping sigma factor. But there are a bunch of different kinds of sigma factors in bacteria. So these are some E. coli sigma factors, as an example. There's sigma A, sigma B, sigma D, sigma H. There are different sigma factors in Escherichia coli. And each one of these different sigma factors bind to different promoter sequences. And I'll show you some examples here on the next slide. But what happens here is they also control genes involved in different responses. So for instance, sigma A is going to bind specific promoter sequences in the front of what is called housekeeping genes, genes that are just always on. But sigma B, it actually binds to promoter sequences that are often involved in stress response genes. And so it might be low nutrient stress, might be high temp stress. Sigma D controls genes. It binds promoters in front of genes that are often involved in motility, movement of one spot to another. Remember when we talked about motility, I said sometimes cells can be motile, sometimes not. They can turn motility on and off. And an alternative sigma factor is going to be involved in that process. Sigma H controls genes involved in starvation, stringent response that we talked about earlier in the semester. What alternative sigma factors are, though, is they control different gene sets. Sigma AB controls genes that are involved in stress response. A bunch of different genes involved in stress response. Sigma H controls genes, a bunch of different genes that are involved in starvation. And by turning the sigma factors on and off, we control all the genes they could control. So we can turn these sigma factors. Different gene sets can be expressed by changing sigma factors. And one thing I want to say is these different sigma factors can be turned on or off. I cannot type. Now, the idea here is if you turn off sigma B, do any of the genes involved in stress response get express them? So by turning off sigma B, you turn off all the stress response genes it controls. By turning on, for instance, sigma H, you turn on all the starvation genes that it controls. So this is a way of coordinately regulating hundreds of genes of like function throughout the entirety of the genome by just turning one sigma factor on or one sigma factor off. Yeah. Here.
With these sigma factors, do they have to bind to specific RNA polymerase cores?
No, they all bind to the same RNA polymerase core.
Okay, so it doesn't really matter which one they
Yeah.
bind to. Okay.
They don't. The RNA polymerase core is always the same. What differs is the sigma factor part. And then also what differs is what promoter they recognize.
Okay.
So in Escherichia coli, sigma A recognizes this promoter. This is the -35 box. This is the -10 box. Sigma B recognizes instead this promoter. You can see it's not the same sequence. Okay, so this is the promoter for sigma D. This is the promoter for sigma H. These are the consensus sequences for those promoters. They're all different sequences that are being recognized then. But this promoter is going to be in front of starvation genes. This promoter is going to be in front of motility genes, genes involved in motility. This promoter is going to be in front of stress response genes, for instance. And then you can turn sigma B on and off. And by turning sigma B on, it turns on any other gene in the entire genome that has this promoter then. A little bit easier to understand if I go through this. This is my bacterial cell. This is its circular chromosome. Each one of my little boxes is a different gene in its gene sets. Okay. Obviously, no way that this is comprehensive. I'm just using this as a select example. Usually bacteria like E. coli have like 5,000 different genes. But let's just say we have a subset. Some of these genes I have highlighted in blue might be housekeeping genes. These are genes that the cell always needs of. Could be some metabolic genes, could be genes that encode for making ribosomes, could be nutrient transporters, cell wall biosynthesis enzymes, RNA polymerase core, could be then some genes involved in cell division, for instance. These might be some examples of what are called the housekeeping genes, the ones that are always on and always present. Sigma A is often always on, always present, often doing its job then. And there's promoters that sigma A recognize in front of all of these genes. So the sigma A promoter is in front of each one of these genes. Sigma A binds to that promoter. Sigma A results in expression of those genes. And my little arrows here are meant to be that they are being expressed into messenger RNA. Okay. Questions about that so far? Well, let's say the cell starts to load on nutrients. So now what happens is the cell is low on nutrients. Well, one of the first things the cell does is, hey, I'm running out of nutrients, but I still have enough energy. They're just starting to run low, but I'm not particularly happy where I am. Grass is always greener on the other side, so let's go try to move to a new spot. So as the cell starts to run low on nutrients, maybe it turns on motility genes. There might be a series of genes. There might be a gene here involved in motility, a couple genes here. There might be an operon here involved in motility. How do I turn on all those motility genes at once? I start making the sigma D for motility. And then sigma D will bind to those promoter sequences. All those motility ones will have this sequence in front of them. They will have that promoter sequence, sigma D will bind there, and you'll get expression of those. But by just turning on sigma D, I also can turn on all the genes it controls and binds in front of. So by turning this on, I can also turn on that gene, and this operon, for instance. And then maybe the cell starts making flagella and becomes motile and swims off to some new location. Maybe it doesn't find any nutrients. So now the cell has not successfully moved to a new spot. It's still running low on nutrients. Maybe it enters into stationary phase because it's starving. Well, then maybe now, because there's starvation conditions, it's starting to make something like, we talked about the stringent response earlier in the semester. Stringent response is response to low nutrients. What was anybody happen to remember what the name of the cellular alarm signal is? The alarmone that controls the stringent response?
PPGPP.
PPGPP. PPGPP starts getting made. I told you PPGPP starts triggering changes in the cellular behavior. How does it do that? It turns on sigma H. And then sigma H comes along, and any gene that has the promoter sequence of this in front of it, sigma H recognizes and binds to. So I have a series of genes here that might then have that sigma H promoter. Here, 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 here. Sigma H binds to those genes and then causes expression of those genes. And then maybe you get things like increased cell wall cross-linking, detoxification enzymes, osmoprotectants, increased nutrient transporters. So you start to turn on stuff that's going to help it survive through those starvation conditions then. But you do this, all you got to do is turn on one single sigma H gene, one single sigma factor, and then that activates all the gene sets it controls. Questions about that? By the way, do eukaryotes do this? Eukaryotes don't quite do this because they don't have sigma factors. And so this is sigma factors and alternative sigma factors is kind of a bacteria or archaea specific version of regulating gene expression. All right. All right, this leads me to an interesting organism for you, and it's one we've worked with in lab quite a bit, Bacillus subtilis. This is a gram-positive bacteria. It lives out in the soil, and it can make endospores. And we're going to talk about that more here in a second. So here's some endospores. It is actually used commercially for a wide variety of purposes. Anyone ever heard of natto before? No, it's fermented soybeans. So this is it right here. It's a fermented food. How do they make fermented soybeans? They actually mix Bacillus subtilis and soybeans, and then they let it set for a couple weeks, and they eat this in Japan. No? Okay. Fermented food's a lot bigger of a thing over in like certain Asian countries. Kimchi. Anybody ever eaten kimchi? Yeah. Sorry. Traditional way to make kimchi is you mix some sugar, salt, and then bacteria into things like cabbage and other vegetables, and then you bury it for 2 weeks, and then you eat it. That's not the way they actually make kimchi that you've probably eaten, though. They speed up the process just by adding acids and some other stuff. instead. So don't worry about it. I'm not trying to turn it off kimchi. It's actually quite delicious. Anyways. All right. We actually use it to make other stuff. So for instance, this is Serenade. If you go into, say, Home Depot or Lowe's or any store, you can actually find this. This is an antifungal spray. So if you have some fungal infections in a garden or out around your house, you might actually throw this. Turns out Bacillus subtilis can kill fungi. And so this is used to treat certain fungal infections of like garden plots and stuff like that. Bacillus subtilis is a pretty powerful genetic system. It's actually naturally competent. What's that mean?
Machinery.
Yeah, it takes up DNA from the environment, and so therefore it's capable of transformation. So that type of horizontal gene transfer we talked about. Sporulation, it also makes endospores. But it's endospores, and the reason I'm bringing it up right now is it's controlled by a series of sigma factors, or what's called a sigma factor cascade. All right, what do I mean by that? If we look at sporulation in Bacillus subtilis, starts off with a mother cell. It will develop into a spore, and then that spore goes through an entire developmental process to make that endospore. That spore, as again, the endospore is metabolically dormant. It's very resistant to harsh conditions of either hot or cold, to desiccation, dry, or even to radiation. So that endospore is really hard to kill. It helps the cell survive through harsh conditions. Turns out sporulation is also controlled by sigma factors. In this case, I might go from a normal healthy cell, but now it starts to go into starvation. Under starvation conditions, sigma H is turned on. Sigma H then controls about 90 different genes involved in starvation responses and surviving through starvation responses. But one of the things that sigma H does is it also activates sigma F in Bacillus. Sigma F then comes along and turns on about 50 different genes that are involved in spore production. So when sigma F is turned on, you start the first processes of making an endospore. And it goes on what are called spore genes. These are in the genes that are involved in the early stages of developing an endospore. Sigma F will turn on these spore genes, but they also then turn on another sigma factor called sigma E. Sigma E comes along, it controls about 270 genes. And it controls some genes in the mother cell, again, that helps to survive it through some harsh conditions, but then it starts to develop the spore a little further as well. And then it activates another sigma factor called sigma G. And sigma G then controls about 90 genes, and it's expressed in the late spore section, and it basically finishes up then the expression of the endospore. Finishes up that endospore structure. Oh, sorry, it activates sigma K. That's it. Finish up the endospore, but then you got also a sigma K will come in, activates about 140 genes in the mother cell. And again, sorry, that will finish up the endospore production. Making of the endospore is fairly labor intensive, fairly expensive. And you have to go through this cascade of sigma factors where sigma H activates this sigma factor. Sigma F then activates sigma E. Sigma E then activates sigma G. Sigma G activates sigma K, which then finishes up the process then. It has to go through this to this to this to this to this. This is called a cascade event. This controls the timing involved. It also creates checkpoints where the spore formation can actually be aborted at any particular case. So if the spore was being produced, say it was this far along the process, but conditions suddenly got better, there was some more, it wasn't in starvation anymore, some new nutrients came along, it would actually stop the entire process after that as well, because it's expensive to do this and it wouldn't be needed any longer. So it provides some control of the system. It also controls some timing to the system. Questions about that? All right, so that's alternative sigma factors. That's not something you guys have probably ever had before, because again, it is a bacterial specific type of process. Questions about that? All right, next type of active regulation that we're going to talk about is some DNA binding proteins that can control gene expression. So transcription in prokaryotes is regulated by a wide variety of DNA binding proteins. DNA binding proteins will, well, bind to DNA sequences. But they bind to special, particular DNA sequences, and usually only under very particular conditions then as well. So these DNA binding proteins will be sequence specific to where they bind, but also conditional, environmental condition specific. Environmental conditions will turn these DNA binding proteins on or off. If the DNA binding proteins bind to the DNA, then they'll affect gene expression. Does this particular gene get transcribed into a messenger RNA and eventually into a protein? So is that gene on or off? However, DNA binding proteins, some kinds of DNA binding proteins can increase gene expression. Some DNA binding proteins will decrease gene expression. It is dependent upon which one we're talking about. And usually what they do is they impact how often or even if RNA polymerase can bind with a promoter. So what they do, a lot of these DNA binding proteins will sometimes help load RNA polymerase onto the promoter, and they might increase expression levels then. Some DNA proteins might actually make it so the RNA polymerase cannot bind to the promoter at all, and then they would turn off gene expression. We're going to go through some different examples of this, but kind of the classical example of gene expression in prokaryotes is the lac operon. The lac operon codes a series of enzymes involved in catabolism of lactose. What do I mean by catabolism of lactose? Yeah, we're going to break lactose down for energy. If we look at these, lac operon is a series of 3 genes: LAC-Z, LAC-Y, LAC-A. What's an operon?
The whole like set of genes.
It's a set of genes that are co-transcribed.
Oh,
And so there is one promoter that's going to be up here that then you make a messenger RNA, and the messenger RNA goes through LAC-Z, through LAC-Y, LAC-A. LAC-Z gene, LAC-Y gene, LAC-A gene are all on the same messenger RNA transcript, or they're what is called co-transcribed together. Or they're all expressed as a unit together in the messenger RNA, is the other way to kind of think of that. LAC-Y codes a lactose permease. This is just a fancy way of saying it transports lactose into the cell. Lactose is then brought in by LAC-Y. It's a type of secondary active transporter, and specifically it's a symporter. It uses proton motive force to bring lactose into the cell as the energy source. So it uses the proton gradient, and you bring in lactose and the protons both in. But the protons are going from high to low concentration. The lactose is going from low to high against its gradient. Lactose is a disaccharide sugar. Lactose is a combination of a glucose sugar plus a galactose sugar. It's this disaccharide. It's a glucose joined to galactose disaccharide. LAC-Z is an enzyme called beta-galactosidase. It's an enzyme, and what LAC-Z does is it breaks the 2 sugars apart. So it takes the lactose, which is a disaccharide, and breaks it into a galactose monosaccharide sugar and a glucose monosaccharide sugar. And those could go into glycolysis, they could go into pentose phosphate pathway, but they're going to use the lactose if it's present. The thing with then the lac operon, promoters, again, mark the start of transcription. So promoters is where we start transcription. The problem with the lac operon is the promoter of the lac operon is a weak promoter. It has a sigma A promoter, but that sigma A, its sequence in front of the consensus sequence for the lac operon promoter is very far away from the consensus sequence for sigma A. So the promoter that's up here is not close to the consensus sequence. In fact, it's quite far away from the consensus sequence. What that means is sigma A doesn't really bind to it. Sigma A does not bind well to it, and therefore you do not get much expression of the lac operon. For the lac operon to be expressed, it requires a transcriptional activator. This is going to be a kind of DNA binding protein. In the case of the lac operon, its activator involves also an operator sequence. So we have operator sequences versus now also promoter sequences. Promoters are where RNA polymerase binds. Operators are DNA sequences that is the binding site for some type of regulatory DNA binding protein. In the case of the lac operon, it has an operator sequence that's in front of it, and it is bound by a protein called CAP, catabolite activator protein. And CAP can bind onto an operator sequence and activate the lac operon transcription. But for CAP to come in and do this, CAP has to be activated. And it requires a small molecule called cyclic AMP, cyclic adenosine monophosphate. And then once you have CAP, cyclic AMP, then it binds onto the operator sequence and it helps load RNA polymerase onto the weak promoter. Okay, so I'm gonna go through that a little bit more step by step. Cyclic AMP is synthesized by an enzyme called adenylate cyclase. Adenylate cyclase will turn ATP, adenosine triphosphate, into cyclic AMP, cyclic adenosine monophosphate. And again, it just looks a little bit like this. Adenylate cyclase will bind to a molecule of ATP, again, ATP, adenosine triphosphate, and it turns it into this, what's called cyclic adenosine monophosphate. It's an adenine nitrogenous base. It's still the ribose sugar, but then you do this weird thing where the phosphate group is simultaneously attached to the 5' carbon and the 3' carbon. And this is the cyclic part. Okay, and you clip 2 of the phosphates off. Questions about that? Cyclic AMP will activate CAP, but specifically it activates CAP because CAP will be bound by cyclic AMP, and this will help then a second copy of CAP come in together. So cyclic AMP helps CAP fold into what is called a CAP dimer. So really, I probably could. Are you guys comfortable with the term dimer? What's di mean? Two. So what's going to end up happening is CAMP binds to CAP. I know, this is horrible. CAMP binds to CAP. That activates CAP and allows it to interact with a second copy of CAP. And then you get a CAP-CAP cyclic AMP dimer. So the dimer just is restoring the fact that you have 2 copies of CAP, and that's what I'm showing you here in this diagram. Once you have 2 copies of CAP, they bind to the operator sequences. So it binds the operator sequences. And do you guys see what it does to the DNA then, though? It bends the DNA. Yeah. So what ends up happening? Here's cap. Here's a second copy of cap. There's one copy in the light blue, one copy of cap in the kind of purplish. They bind onto specific operator sequences, but when they do that, they bend the DNA. And when they bend the DNA, it actually makes it so that the RNA polymerase binds better onto that weak promoter. So the cap cyclic AMP complex, they make it so that the RNA polymerase does a better job of recognizing that weak promoter and binds to it much better than it would without cap present. So instead of having that weak promoter, by bending the DNA just a little bit, it makes that promoter look better to the RNA polymerase inside. RNA polymerase comes on more often. RNA polymerase activates transcription of that LAC operon. So just as a kind of a summary of that, the promoter for LAC is very far away from consensus sequence. So it's a very weak promoter. It's barely expressed whatsoever. This means to express the LAC operon, you require an activator. The activator is the cap cyclic AMP complex. Set cap and cyclic AMP will bind to the operator. This bends the DNA a little bit, and it stabilizes the ability of an RNA polymerase to bind to that weak promoter. It basically turns that weak promoter into a regular promoter. When you bend that DNA just a little bit, it makes the promoter and the RNA polymerase recognize one another better. Well, it makes the RNA polymerase recognize that promoter better, and therefore you get transcription. Questions about that? Now, the thing about all of this is the bacteria also have food preferences. For instance, glucose is preferred food source compared to lactose. Why? You don't have to digest it apart. There's an extra, lactose is a disaccharide, there's an extra digestion step. And it turns out that extra digestion step costs some energy. So if you have both glucose and lactose around, it's cheaper to just eat glucose. Does that make sense? Okay. That's not to say that lactose is a bad food source, just glucose is better. And as long as there's glucose around, it's cheaper to only eat the glucose as a sugar source. As a result of this, though, since it's cheaper, it's more efficient to eat glucose. The presence of glucose will actually deactivate the LAC operon. And it turns out glucose inhibits adenylate cyclase. So this adenylate cyclase, what does adenylate cyclase make again? Cyclic AMP, CAMP. So glucose is around in the cell, turns off the adenylate cyclase enzyme. You do not get cyclic AMP level. AMP me, and therefore, in the presence of glucose, cyclic AMP levels drop. If cyclic AMP drops, can CAP work? No. Without cyclic AMP, CAP no longer activates the LAC operon, and then the LAC operon is not being expressed. You see in my diagram here how I don't have any messenger RNA being made? It means it's not going to be expressed. This is referred to as catabolite repression. The glucose is the preferred catabolite, and its presence will turn off the usage of the lactose as a catabolite, as an energy molecule. Questions about that? That is the LAC operon, but I'm using that as an example of DNA binding proteins. Activators. When I turn the term activators, these are DNA binding proteins that bind to specific DNA sequences and activate expression of the downstream genes. Whatever they bind to in the DNA, the sequence they bind to in the DNA is called an operator sequence. And typically activators help load RNA polymerase onto a promoter. They stabilize the RNA polymerase promoter interaction, and therefore you get increased transcription, increased gene expression. Again, I was using CAP as an example of an activator, but there's a bunch of different activators for a bunch of different genes in bacteria as well. So this is the more in general what happens type of thing. Questions about that? Here's the thing about the lac operon. It can be activated, but why do we often use lac operon as a classical example of gene regulation? Because it turns out the lac operon can be both either activated sometimes, but also repressed or turned off in other kinds. So the lac operon is dedicated to lactose catabolism. In the absence of lactose, would we want to express the genes in the lac operon? No, because that would be a waste of resources. So if there is no lactose around, if lactose is absent, expression of the lac operon wastes cellular resources, and cells never want to waste resources. As such, the lac operon is repressed when lactose is absent, and it's only expressed when lactose is present, the flip side of that. So how does this part of the system work? There is a gene somewhere in the genome, has a LAC-I gene. LAC-I codes for a protein called also LAC-I. Do you guys remember when I said gene sequences are typically lowercase and italicized, whereas protein sequences are typically uppercase, not italicized. So this is the LAC-I gene in the DNA that codes for the LAC-I protein, an enzyme. Okay. LAC-I is a repressor of the lac operon. It binds to operator sequences, different operator sequences, though. You guys see how I turned it from green to red operator sequences? Also, CAP operator sequences, where are they relative to this is the start of transcription in a little diagram. Where are the operator sequences for CAP? Further upstream. Where are the operator sequences for LAC-I?
Further downstream, but
Oh,
not as upstream as the other one.
actually, they straddle it.
Oh.
There's one in front and one behind the start of transcription. They straddle the start of transcription and they straddle the promoter sequence as well. So LAC-I is a repressor and it binds to operator sequences. And when it binds to the operator sequences are different sequences in a different location. But when they bind to this specific operator sequences, it blocks the expression of the operon and turns it completely off. This is LAC-I, a couple copies of it bind. And then what's it do to the DNA? It loops it. And what is part of the loop? The promoter. And do you think if the promoter is doing this weird bent loop, do you think RNA polymerase can bind to it, recognize it, and bind to it properly? It can't. So when LAC-I binds to an operator sequence, it binds to one here and one here. These are the ones in red. And then it loops the DNA in between, and it makes the promoter sequence completely invisible as far as RNA polymerase is concerned. And that turns off transcription. It's actually kind of neat. Now, obviously, I got to make it a little bit more complicated than even that, and it's already complicated. LAC-I, its activity is turned on and off. So, whoops. When the inducer, lactose, or sometimes also referred to as allolactose, when lactose is present, lactose will bind to LAC-I and inhibit it, turns LAC-I off. So if lactose is present, this is my lactose, it binds to LAC-I and makes it so it cannot bind to DNA. It prevents its DNA binding activity of that repressor protein, and thus it results in de-repression of the lac operon. This is hard. Why am I using the term de-repression specifically? If LAC-I can't bind, what's LAC-I's job when it's bound to the DNA?
To repress that site.
It represses it and turns it off. Now, let's say lactose is present. It results in the LAC-I repressor being inhibited. This will result in de-repression of the lac operon, but does it necessarily result in expression or activation of the lac operon? No, it doesn't turn the lac operon on. It just means that it's possible to express it or it's not being repressed. This is called de-repression. But remember, the lac operon has a weak promoter, and it also needs cap protein to bind to be expressed. Okay, so I'm going to go through this a little bit more in summary here. But what is basically happened is lactose will permit the expression of the operon that is then needed to consume it, is the idea here. Okay. First, in general, I'm going to come back to this, what's coming over here more systematically. But first, in general, LAC-I is an example of a repressor protein. Repressors are DNA binding proteins that bind to specific DNA sequences and then inhibit gene transcription. So they turn it off. The DNA binding site they bind to is called an operator. DNA binding proteins bind to operator sequences. But there are some DNA binding proteins that bind to an operator and then act as an activator. There are some DNA binding proteins that bind to an operator and then act as a repressor. Okay. Typically, in the case of repressors, their binding overlaps the promoter in some way and then blocks the ability of RNA polymerase to bind to it. In essence, most repressors hide the promoter. So RNA polymerase can't bind to it, and they turn off the gene. Questions about this? This is more the general how repressor proteins work in general. All right, let me come back then, and the final thing for today is let me come back to the lac operon expression more holistically. If you have no sugars in the media, here's what happens. Without glucose, adenylate cyclase works, cyclic AMP is made, cap protein is activated, and cap binds. But without lactose, LAC-I also binds, and LAC-I activity is dominant. The repressor has a dominance in this particular scenario. So if both cap and LAC-I are going to bind when there's no sugars in the present, but because LAC-I is dominant, lac operon's off. If glucose and only glucose is present, glucose inhibits adenylate cyclase, no cyclic AMP gets made, cap protein is off, cap doesn't bind. But also there's no lactose in the media, so LAC-I is active, LAC-I binds, and it's really off. Okay, there's no activator and there's the repressor. So it's very, very, very off. They're still off. If there's both glucose and lactose present in the media, then what ends up happening is glucose inhibits adenylate cyclase, no cyclic AMP is made, cap is not active, cap does not bind. But lactose binds to LAC-I, inhibits the activity of LAC-I, so it cannot bind and is also not bound. So in this particular case, I'm not repressing, but I'm also not activating. And then you get off to maybe very weak expression. I probably should put very weak expression here. Very, very weak, just a little bit. When you have lactose and only lactose present, lactose inhibits the activity of LAC-I. LAC-I doesn't bind. So we get de-repression. But also, there's no glucose around. Without glucose, adenylate cyclase is active. Cyclic AMP gets made. Cyclic AMP binds to cap. Cap is activated. Cap binds. Cap activates the expression. And in this particular case, we have activation and no repression. and we get expression of the lactoferrin when lactose is present and only lactose is present. Questions about that? I know that's hard. We're going to stop there. I went a little over. We're going to stop there, but I'll talk about a little bit more about catabolite repression. But then we're going to talk about some other types of regulation, which will go a little quicker. We'll probably finish up most of this on Monday, then. Both the other types of DNA binding proteins, but also the translational regulators.